you also tell me, all of you who are answering and who are so engaged, and other people as well, who, if you're, I mean, I hope you become interested as well. Can you also tell me, this personalized medicine, what does that mean to you? Is it only for a single individual, or do you think it could be for, say, something else? So when I use the word personalized medicine, all of you are here in this FDP on tailored treatment and advances in personalized medicine. Personalized na enna? ஒரே ஒரு individual, based on individual cell type. Okay. Anyone else? Individuals or a community? Okay, good. For everyone. All right. Thank you, Mr. Jayamani Kandan, Mr. Balakrishna Raja, Mr. Saravanan. Munishwari, Ms. Munishwari says a specific drug for specific disease for everyone. Okay. So this is going to be including our surroundings, all right, customized, Mr. Kumaravel, okay. Excellent, based on individual. So I think all of you are getting the sort of this thing that it could be for an individual, but it could also be for a group of individuals, right? And a lot of my work today and a lot of my talk today is going to be looking at communities, and not communities based on specific genotypic shared sort of genotypic consistencies. These are actually communities based on geographical proximity. So you and your neighbor and that person's neighbor may not be genetically similar at all, but you are geographically proximal to each other, and your environments are the same. So automatically you are, of course, this is obvious to anybody who lives. If you are in a particular community with common air, water, soil, then you're automatically sort of both contributing to that environment, and that environment is also affecting you in positive or negative ways, right? So while most of tailored treatment and personalized medicine may talk about a single individual person, this perspective that I'm bringing today is going to be actually about the environment around a person and how that can be harnessed to look at individual or a group of people's treatment. And hopefully that doesn't seem as far-fetched to you by the end of the talk. Can we go to the next slide, please? So these are the objectives for the session. I'm going to be discussing the work that both our group as well as other groups that we work with and other people whom we don't work with, but in the country and in the world, have done in tracking water-based cues for public health. What are the signs that we get in the water that it could be wastewater, it could be other types of water. What are the signs that we get that can help us in infectious diseases surveillance? And then once you have some degree of data on that surveillance, how does that help you in public health action? So basically, how does that help you respond? Ultimately, we would like to be able to say that we have controlled a particular disease in certain ways. One of the things that's very good about Tamil Nadu is its very strong public health system. Many public health people whom I speak to in Tamil Nadu themselves will be like, "Avlo onnu nanna illa sir or madam, edho pannittu irukom paravailla." ஆனா நீங்க தமிழ்நாடுல கம்பேர் பண்ணீங்களா, கர்நாடகாவோடய கம்பேர் பண்ணீங்களா, இல்ல if you compare it to north of India, Tamil Nadu has excellent public health systems, and a lot of that is because of very good surveillance systems. Surveillance na enna? இதே மாதிரி ஒரு கம்யூனிட்டில what is happening in terms of not just disease, but it's also in terms of risk factors. இந்த இடத்துல நிறைய factories இருக்கு, நிறைய air pollution இருக்கு. So that itself is part of surveillance. வேற இடத்துல இந்த group of workers, they are at high risk for these kind of health problems. That is surveillance. From COVID, all of us have undergone that experience with COVID, And we all know what kind of surveillance the government put in place. யாருக்காவது RT-PCR result, நீங்க RT-PCR பண்ணீங்கன்னா அந்த ரிசல்ட் வரும் உங்களுக்கு. And if you're positive, then you get a call from your city or your community or sometimes even the state saying, எப்படி இருக்கீங்கோ, do you need access to a hospital, that kind of stuff, right? All those are parts of surveillance. I'll also describe the insights gained from mega scale rejuvenation of lakes with a focus on AMR. So we speak when typically all of you are from what kind of background? Are you all from health background? Are you all from life sciences background? What kind of background are you all from? Life sciences, Hinduja, okay. Okay, Mr. T says life sciences. All of you are okay. Biotechnology, life sciences, okay. Excellent. So this does look like a mostly biotech life science background. Great. So all of you also, because you're all faculty, I'm sure you must have heard of antimicrobial resistance. Basically, when a particular pathogen no longer responds to an antibiotic or an antimicrobial, right? So we hear about this from a biomedical background. Doctors will talk about AMR, veterinarians will talk about AMR. Of course, biotechnologists do talk about AMR a lot because the drug discovery is such an integral part of biotechnology. It's such a big component of any kind of biotechnology undergrad or a postgrad course. But have you ever thought about AMR from the point of view of geology or geography or hydrology or urban planning? So a lot of the work now is now becoming saying, where is this AMR actually coming from? Is it just because of antibiotic use, or is it actually in the environment around us? And if it's in the environment around us, it's not just life sciences and medicine that contributes to AMR, but it's also a good amount of your environment, water, air, soil, all that stuff. And we'll attempt to connect all these things. All of us, when we talk about science, we talk about science in a lab or in a hospital. But in real world, it's very different. And actually, all these things, if you want to see real life change in a society, you have to bring in your real life messiness into understanding scientific problems. All right. Okay. Next slide, please. So I'll briefly talk about myself and my company, because I'm not sure how many of you understand the perspective I'm coming from, and that will help you understand why I ask the questions I ask. What are the questions that drive us? I'll tell you a little bit about the work that we've done in wastewater surveillance for diseases, a little bit about AMR and antimicrobial resistance, and a little bit of the real world as where all these things kind of really merge. Next slide, please. But before I get to the meat of the matter, these are the questions I'm going to be asking you at the end of my talk. So these are all questions that I would like you to think about as you listen to my talk, because you will be asked the same questions maybe about 15 minutes from now. So my question for you is, how would you think through the ethics of urban wastewater for rural use? சென்னையில ப்ரொடியூஸ் பண்ற சுவேஜ், அழுக்கு தண்ணி என்றால என்ன சொல்றா, கட்டர்ல இருக்கிற தண்ணி, அது வந்து if you send it to a village or to a smaller town, தஞ்சாவூருக்கே அனுப்பலாம்னு நினைச்சுக்கோங்க, because I know there's somebody from Tanjavur here. If you were to get treated wastewater from Chennai, what would you think about the ethics of that? Is that the right thing to do or the wrong thing to do? So think about that. What are ways by which wastewater used would become acceptable to you personally as an individual? Would you be willing to use wastewater for agriculture? Would you be willing to use it for your pets, for your cats or your dogs or cows? Would you be willing to use it for your drinking water? Would you be willing to drink that water? So these are the questions I want you to think about. And finally, I would like you to think about what worries you about antimicrobial resistance. Okay. Next question, please. Next slide, please. This is me. Like I told you, I'm Varsha Shridhar. I started off as an industrial biotechnologist. In fact, Dr. Gauri Shankar, who is the main leader of this course, and I were classmates in Anna University in our BTech program. After that, I did a PhD in molecular microbiology and molecular virology for 10 years in the US. I came back to India also about 10 years ago and started my own company. So when I started the company, it was an R&D company, and during COVID, I really sort of got interested and very deeply involved in public health. So now I call myself a public health practitioner, and I focus on primary care diagnostic systems. And this is the name of my company. Next slide, please. So this is what our company has been doing for the past, 9 years that we have been alive. The company was Molecular Solutions, MSCH, was started in 2016. We started off as an R&D and innovation lab. During COVID, we became Bangalore's first COVID testing center in a primary healthcare clinic. Usually we had all these testing centers in big hospitals. Now we basically set it up in a PHC. And then we started work initially. Sorry, I don't know if you can hear me. Accidentally got another call. Yeah, there we go. Then we started work on wastewater surveillance. The reason I started this was because this was during lockdown. There is a Mori right behind my house. And we had read some papers that in Amsterdam, in Netherlands, one group had found COVID in the wastewater 4 days before the first set of people in the entire country became sick or showed signs of clinical SARS-CoV-2 infection. So they said basically this wastewater could be a potential early warning. So we wanted to try that. We tried that, and we got very, very nice results with that. And that's why we were able to expand on it. So for the next 2 years, we basically established this precision pandemic health platform, which was a citywide wastewater surveillance platform. It covered 11.5 million people, and we were able to show hotspots of COVID before people started becoming sick. And along the way, because we started this thing with diagnostics, we scaled that also. We did more than 1.5 million COVID tests and all that stuff. So anyway, lots of scale. After COVID, obviously there was no need for that kind of scale. Reduced it to 3 main verticals: clinical infectious diseases and microbiology. So we work a lot with hospitals on AMR. Primary care diagnostics. How do you ensure that diagnostics reaches the person who's going to a primary healthcare center or a village clinic somewhere, so that we are not constantly coming back for tests? And environmental surveillance in general. Okay, next slide, please. And that's what we are doing over the past couple of years. These are the questions that drive me and my team. இங்க யாருக்காவது உங்களுக்கு ஏதானும் experience இருக்கா இதே மாதிரி? If anybody in your family or anybody has had TB, for example, or any kind of disease where you don't immediately get a lab report? As there, let me go back to the chat section to see how many people answer. In India, rules are not that strict, especially in urban. Any water must be treated for agriculture. Yeah, we can talk about these questions in a little bit. But has anybody had the experience of trying to get a lab test and not actually getting it immediately, and you have to keep coming back over and over again? Okay. If you've not, that's great. Maybe again, it has to do with Chennai and Tamil Nadu and its general these things. But usually, for the most part of India, including, I would say, in a large part of Karnataka, what typically happens is if somebody falls sick, the first thing they do is buy drugs over the counter. And if that doesn't help, they go to a doctor. And then if you go to the right-hand side of this golu circle figure, what happens then? The person gets referred for lab testing. You could basically lose that patient to follow up over here. Many times they have to search and locate the labs, especially if it's not an easy test. If it's not like a CBC or an ESR, but let's say it's a sputum CBNAT. In the past 2 years, it might be easier, but definitely about 2 years ago, it was very difficult to get a lab. You had to search and find a lab that would do the CBNAT for you. you'd have to produce the right kind of sputum, you'd have to collect the lab report, you'd have to act on the lab report. So you can see it's a long journey from the time a person feels sick till the time they are cured, or at least get accurate lab information and healthcare tests. It is not an easy process. There are many times when people can just get overwhelmed by the healthcare system and say, I can't do this anymore. I would much rather, go back to work and somehow struggle with this and find out what's really this thing. So my question was, how do we ensure access to all for timely and appropriate healthcare? We go to the next slide. This is a slide from a real life situation from Canada, where they actually have incredibly good healthcare. So if you have a community of 1,000 people, maybe 800 people will report symptoms, which means they actually have clinical signs of a particular illness. Of them, less than half will consider seeking medical care. Of them, even fewer will actually go to a proper physician's office. There'll be many people who visit complementary or alternative medical care providers, etc., etc. Less than one of those 1,000 people will actually be admitted to an academic medical center. Just click the slide just now. There's an animation that should come up. If you think about it, and all of you who are in life sciences, reading papers on infectious diseases will know this. Nearly all our understanding of disease, of any kind of disease, is based on the data that comes out of an academic medical center. A tertiary care medical center only sees a fraction of the people who are truly sick, but all our data comes from there. All our public health systems are based on the data that depend on somebody going to a medical system. So all are in green, in this green circle, people who end up in an emergency department in a hospital, people who are hospitalized, people who are in a tertiary care system. This is the data that feeds into all our public health systems, right? So how do we go about trying to be proactive about disease control instead of waiting for people to fall sick enough to get our attention? If you who will end up in a primary care center or a hospital? Someone who's really sick. But there must be a whole lot of people, but by that time the disease is already come up to a high enough level that you're not able to actually, you're always going to be 2 steps behind the disease. It's already made someone sick enough. That person has already infected somebody else. And then the public health response is like, oh, hang on, what's going on, right? We're always 2 steps behind. So how do we become proactive about this? Next slide, please. So this is a little video. I don't know if you can play it. Whoever is controlling the video, can you click on it and see if that plays? Yeah. And you can increase the volume. Can you increase the volume? I can't hear the volume. Okay, I guess the volume is not there, but you can see the subtitles. It works beautifully in lower income sections of urban areas where people's poop converges. It's the first time that we see something that belongs to everyone, and people can actually protect themselves. Environmental surveillance has a huge potential because there are so many people who don't have access to regular testing. Poop is a great equalizer. Everybody poops, whether they're rich, they're poor. It is not just important for India, but for everyone. To solve the world's most pressing problems, we need to trust those closest to the challenge. These are the solutions that make our world a better place for everyone. This is Rethinking Possible. For anybody who walks around Bangalore, the first thing that you do see are the open drains. I don't think there are too many high-income countries with open drains. I would be very surprised, in fact. I'm Varsha Shridhar. I am a molecular biologist and a biotechnologist. In the 60s, polio wastewater surveillance was basically the first large-scale public health wastewater surveillance. But after that, there hasn't really been any sort of ongoing research into what can wastewater be used to test for.
It wasn't until the urgency of the COVID-19 pandemic that interest in wastewater surveillance renewed. And after one study showed that underground wastewater surveillance could serve as an early warning system, it prompted the question, could it work in open drains too?
My name is Dr. Angela Chaudhary. I serve as Chief Catalyst of Swasti. It's a global public health organization that serves some of the most marginalized populations. In March 2020, already many people were affected by COVID. We really wanted to know if open drains held some clues for us. So we had to prove for ourselves that this experiment would actually work.
I would not say we were winging it, but it was definitely not something that we had tested before.
We literally had a fishing rope with a plastic bottle tied at the end, and we just kept doing that.
We were just like, put the bucket into the water and carry it to the lab. But when we saw that first positive, I think we both freaked out. There was this kind of shriek, it's positive.
With the backing of Swasti and a host of other health-interested collaborators, it created the Precision Health Platform, a public dashboard where wastewater test results could be viewed in real time and could inform communities to know an outbreak was imminent 7 days before hospitals started filling up.
This meant that government administrators could warn the hospitals. They could plan human resources, they could plan their staff, and they could plan for hospital beds.
Initially, I was not able to see the connection between what I thought would be an isolated research finding to a public health response that could actually lead to some kind of an actionable insight.
The moment there's a spike, our data was better than the...
Angela is an extremely experienced public health professional, and so she was able to see the next steps forward.
It all starts here, with the sewage collection. Through barcodes, they track where each sample is scooped up.
All these sites are geotagged. This is the bottle of the sample which was taken from this particular site. This represents the population of around 500 households on average.
Samples are then taken to Dr. Varsha's lab, where RNA is extracted and tested for various pathogens. Then that information is transmitted to the dashboard, corresponding to each 500 household section of the city. It puts everyone, no matter status or wealth or location, on a level playing field.
It's a lot about governance. Why shouldn't we have access to information like that? The platform itself is setting new standards in what should be a public good. I would like to know what risks are out there in a way that I understand so I can take care of myself.
People who live in slums in India often don't have access to proper health care. So with this information available to community leaders, health authorities, and journalists, it can be disseminated throughout communities, delivering on this massive public health challenge.
If they don't fall sick because they're protected by masks and other interventions, then they're able to lead their livelihoods without any concerns.
What I'm most excited about the future is the integration of environmental signals with community clinical data for conditions like antimicrobial resistance, tuberculosis, and new and emerging pathogens.
In addition to expanding their testing to other pathogens or compounds of public health interest, the Precision Health Platform is also expanding across India. Their framework is available for any place with open drains.
We started with Bangalore, now we're in 5 cities across India, with interests from Cambodia and Philippines as well. Our dream is to see this scale up in as many cities as possible so that the cities are better prepared.
The years since 2020 have only sort of underscored that we are a bunch of slightly weird people who like to test gross things. I think what sort of drives me in this is the constant need for tinkering. We have evolved from a biomedical lab into a public health action on the ground.
Scientists, people who hold political posts, artists, all of these people came together to deliver health outcomes for some of the most marginalized people. Precision Health Platform has shown me local innovations can help us plan and prepare for the next pandemic, perhaps even the next disasters that are going to hit us.
Okay, so I actually asked to pause after the first couple of minutes. So sorry, guys, this went on for much longer than I thought it would. So any questions or comments so far? Okay, maybe not. Let's move on. Next slide, please. So yeah, this is a Precision Pandemic Health Initiative. It's been published a lot. And also, in case any of you are interested in doing this, the protocols for this are available online. So you can just reach out to Dr. Gauri, who can connect with me, and I can send you all the links. Next slide, please. So this is basically what I meant by saying it is personalized for communities. So this is, for example, a graph showing you sort of hotspots of, as Shirish said in the video, about 500 households. So you can say in proximity to these particular places, this is where, say, viral load of a particular infection is going up or going down and things like that. On the left over here is the map of Bangalore, but with hotspots on antimicrobial resistance. And I'll come to that a little bit later. So wastewater surveillance provides community level hyperlocal insights, which can be put into practice. And it's not just a research project, but it's actually it can help a public health system respond to what is actually what is required for that particular community. Next slide, please. So yeah, we did this in a bunch of other infections as well. You can see COVID is marked over here, but we also did it in influenza, and then we did it in RSV and all that. Next slide, please. And this is basically how the early warning system works. Interestingly enough, a lot of the viruses that are actually respiratory also infect the gastrointestinal tract. So this you see, of course, in COVID, because you remember in the early days of COVID, people would say you lose your sense of taste and smell, but you also might get diarrhea. So basically, it is the virus infecting cells of the gastrointestinal system, which is the reason why you get diarrhea, right? And it goes through the wastewater system into your open drains or your sewage treatment plants and all that stuff. But it's not just SARS-CoV-2. Influenza does this, adenovirus does this. A lot of infections, basically, especially viruses, not so much bacteria, because they are a little more restricted in their host receptors. But many viruses have the potential to infect multiple types of cells, just because their receptors are a little more broader in terms of where they are found. Okay, so what you basically see is that the virus is replicating in somebody's body, and it gets shed through stool. We are talking about stool here, but it can also get shed through urine in some cases. And only when the virus particles or the viral replication builds up in a person's body do they actually fall clinically sick. But because the virus is constantly getting shed in the stool, you can actually start picking up these signals a few days before somebody shows clinical symptoms. So you actually see the early warning. That's the point number B over here. Before the clinical cases start rising, that's point number C. So B comes a little bit before C, and so you have a bit of an early warning advantage. Next slide, please. This is data from our own experience in COVID, showing that we were able to show the third wave like 5 or 6 days before the clinical peak went up. Next slide, please. And this is showing the congruence between open drains and underwater STPs, sewage pipes. Because, in Chennai also, I know this is true. I'm assuming this is true in other cities as well. Now, with a lot of interest in infection control, a lot of cities are investing in creating underground drainage. So does what we see in the open drain actually translate to underground drainage? It seems like it does. So next slide. So even if a city increases its infrastructure and becomes a little more, it becomes better at covering up its sewage pipes so that they are actually underground, this would actually work. Next slide, please. So yeah, this is the first story. Wastewater surveillance for SARS-CoV-2 was able to detect hotspots before clinical cases. Now, how do you actually ensure that it works for other infections? You need to get that kind of clinical information. So validation for WWS for other infections, for example, like flu or metapneumovirus, for example, that was such a huge big deal just 2 weeks ago. If you don't have clinical data on this, it's very hard to validate it. You can say, look, I'm seeing this particular infection go up, but would anybody take you seriously if it has not been validated for that infection? Hard to say. So you need to have that kind of clinical information before you can validate the use of wastewater surveillance for other infections. Next slide, please. So actually, before I go to AMR, let me just check the chat session a bit to see if there are any questions. Hello, Dr. Prabhi. Does anyone have any questions? You don't have to put it on the chat. You can just ask me verbally. Any thoughts or any comments? Okay. Shall we go on then? Okay, if you're looking at this on your phone, I'd like you to kind of zoom in to the right-hand side panel over here. This is the test report from our own lab about a few months ago. As you can see, this is a 7-month-old baby, a male child. This came in October, end of October of last year. And this was a urine culture and sensitivity. So basically, this child had been admitted to the ICU for a urinary tract infection. And you can see, basically, that urine grew E. coli, which is a very common this thing, but it says Escherichia coli CRE. What does CRE mean? There are these classes of antibiotics called carbapenems, which are only given to people when the pathogen is resistant to multiple lower classes. Carbapenem are really, when you think about it, among the highest levels of antibiotics that can be given to for a particular infection. You start off with your penicillins and your regular beta-lactams, you kind of go, or to your cephalosporins. Those who teach pharmacology in biotechnology will know this better than me. But basically, you sort of, you have your cephalosporins, or maybe your if the cephalosporins are resistant, then you try fluoroquinolones. If those are resistant, you try something else. You say beta-lactam, beta-lactamase inhibitor combinations. And if those are also resistant, then you go to carbapenems. These are only given, for the most part, in the ICU through intravenous injection. Why should the 7-month-old child, who presumably, you don't expect a 7-month-old baby to be exposed to antibiotics, that he has been infected with an E. coli, which is carbapenem resistant? The child, that E. coli is sensitive to 2 antibiotics. When you see this, it's sensitive to fosfomycin and tigecycline, resistant to everything else, right? Neither fosfomycin nor tigecycline can be given in a child this young. You have the minimum age for tigecycline is 12 years of age. So what do you do in a child like this when this child is so sick? So imagine this to be the case across the globe. When you have young children or even adults who've not been exposed to antibiotics coming to the hospital with antibiotic resistant infections, where are they getting this antibiotic resistance from? Next slide, please. So the AMR that we are seeing across the globe is way beyond the hospital and clinical use right now. Okay, so if you look at the antibiotic use that is going on in our societies right now, 30% of antibiotics only are used in humans. 70% of antibiotics are actually used in animals. And antibiotic usage in animals, not only is it expected to jump 67% by 2030, as you can see on this thing, but antibiotic usage in animals, they are not using this to prevent infections. They are using it in animals to as growth promoters. What somebody found out in the 70s is that if you give antibiotics to your chickens or your pigs or whatever else, think about cattle, or goats, the weight of that animal increases. So basically, then you obviously get more money per animal because the weight is higher, and so etc. etc. So 70% of the antibiotics are used by animals, but not all of these antibiotics are actually absorbed either by humans or by the animals. If you click, I'll just click, no? Wait. Not that. I think you clicked on the link. Can you go back to my presentation? Just click somewhere else. Okay, so yeah, just wanted to highlight that 70% of antibiotics are used by animals or used in the animal industry. And click again. Just want to highlight one couple more things. So, go back. Yeah, just there. That's all. Just stay there. So, up to 80%, not all these antibiotics that either humans or animals are consuming actually gets degraded inside the body. 80% of consumed antibiotics are excreted as they are, as active components in the urine or feces of the animals or the humans who are taking them. Okay? So now what happens to them? Basically, all these antibiotics are landing up in the wastewater. And what happens to wastewater? Some wastewater, sure, are getting treated in your STPs. Places like Chennai, places like Bangalore, I think about 2/3 of wastewater actually get treated. Unfortunately, the amount of wastewater that's being produced by most cities in India is way larger than the amount of STPs that are there, sewage treatment plants that are there in the cities. So you actually have a large amount of wastewater being released without treatment. So where do they go? They go to all our lakes, they go to our rivers. And that same water that's going into our lakes and our rivers is actually being used for agricultural use. It's actually being used for irrigation of our fields and even for animals to go and drink. So while these over time, from our pharmacokinetics in biotechnology, we know that active components eventually sort of decompose, they degrade, right? But the half-life of these antibiotics vary, and they also depend on the amount of environmental exposure they have. So for example, some antibiotics degrade very fast in the presence of sunlight. So you can say, okay, let's keep the wastewater out in the sunlight, let the sunlight, hit on them, and they'll basically split apart into inactive components. But many antimicrobial agents do not actually split apart. They are present in low concentrations. They are not high enough to kill bacteria that are there in the wastewater, but they are low enough to induce antimicrobial resistance in them. So there's this constant evolutionary pressure on these bacteria in the wastewater to become antibiotic resistant because of the low levels of antibiotic that's always in that wastewater. Okay, how does that make you feel? Any questions? I'm not seeing any questions here yet, only about this thing. So guys, I'm not going to keep checking the chat thing over and over again. If you have any questions, please just ask it, okay? Or comments. Next question, please. Next slide, please. So yeah, let me just tell you the story of Bangalore city and its as an urban area and the rural areas. Remember the first question I asked you all was to find out how many of you were from Chennai and how many of you were from places which are not as large as Chennai, right? Even if you're all living in Chennai right now, I assume that many of you will actually have your hometowns in smaller areas. So, for example, my hometown is a smaller part, like what I consider my hometown now at least, is a smaller part, a city called Kolar, which is about 2 hours from Bangalore, right? So if you click on this slide, okay, so a lot of you know about Bangalore. It is considered to be the fastest growing city in the world. We basically doubled our population in 5 years. It is, honestly, it's a little insane. We have about 13.5 million people. It has a very large density of industries. Yes, right now it has lovely weather for now. This is rapidly changing with climate change. Okay, next slide. Here is Bangalore, all the way in the southern, south sort of eastern part of Karnataka, and that is what I have circled in blue. But you can see all the surrounding districts of Bangalore are red. So nearly every district surrounding Bangalore is extremely water scarce. It has very little, all these districts have very little water. The state in general, Karnataka state, even though this may not seem like it to Tamil Nadu, especially given all the water issues that we have, all the water fights that we have between those two states, the state actually has 16 of the 24 most drought-prone districts in India, and nearly 90% of the population is semi-arid and it's dependent on agriculture. Next slide, please. So about 5 or 6 years ago, because of this increasing lack of water, especially around Bangalore, as Bangalore exploded and it keeps exploding in its population, the water resources from surrounding areas of Bangalore are basically being completely depleted. And a lot of these districts surrounding Bangalore have basically completely become zero in their groundwater resources. So about 5 years ago, the government decided that they are going to use treated wastewater from Bangalore to pump into the lakes in all the areas surrounding Bangalore. So this was going to be the big lake rejuvenation project. It's called Kere Sanjeevani. Kere means lake in Kannada. Sanjeevani, of course, is rejuvenation, revival, etc. And these are all the districts that were supposed to get water initially. So there's Bangalore in the center. If you go to the right panel of this slide, there's Bangalore in the center. To the north, to the east, and to the southwest, these are all the lakes and the reservoirs that get this water. And you can see ML, or if you go to the left-hand panel over here, these are the this is the amount of water that is being sourced by the river and where there is a deficit. MLD means million liters daily. So you can imagine the insane scale of this. 1,640 million liters of water. Million liters is the quantity of wastewater generated. So every single day, 1,640 million liters are being generated as wastewater. And of this, 5 years ago, they said we will pump 770 million liters. Now this has gradually increased to over 1,000 million liters. All this water is being pumped to these neighboring districts, Kolar, Chikbalapur, and Anikal, for agricultural reuse through groundwater recharge. Groundwater recharge basically means you pump this into dried up lakes, and then you wait for that water to percolate down into the ground, and then you pump it not from the lake, but from wells and from bore wells in surrounding areas. So you have an extra additional layer of nature itself being a way of purifying that water. Next slide, please. Yeah, so our context for the study was Chikbalapur, which is a district north of Bangalore. So Chikbalapur is located in the southeastern part of Karnataka. It has faced 9 to 11 years of drought out of the past 15 years. It is considered one of 6 permanently drought-prone areas in the country. So if you look at the right-hand panel, Bangalore is here in lavender. Any of you who's taken a flight to Bangalore, you would have reached the airport, which is in Devanhalli, which is given in the green zone. And then above that in yellow is Chikbalapur. And all these little red dots are the lakes that we tested. Well, that was the lakes that we had tested originally, but in this particular study, I'll only be talking about 4 of these lakes. Next slide. This is basically how the area looks. On the left-hand panel is this particular lake in 2018. This is the lake in 2022. You can definitely see there's more green, right? So a lot of there's been a lot of rejuvenation in terms of agriculture. Next slide. These are the 4 lakes that we selected for the study. Next slide. These are the dashboards that we created So you can see how you can basically just look at green space. In at the bottom, you have this extremely dry land. And then 5 years after or 3 years after the the wastewater was pumped back in, you can see there's definitely more green and a lot more agricultural areas that have been revived, so to speak. Next slide. And this dashboard that we created looks at multiple different variants. You can select the lake of interest. You can select which antibiotic or antifungal you want. You can also select attributes of water quality itself, like alkalinity, heavy metals, coliforms, dissolved oxygens, etc. And you can basically pick whether you want the data in the lake or in an open well nearby, which is being used by a farmer, or in a bore well, which is being used by either a farmer or a person who's living there, who's using that water for their own purposes. Next slide. So this is an example. For example, in Bagalur Lake, we want to look at alkalinity. And these were all the points across Bagalur Lake that we tested, all these little pulleys around the lake. And you can check at the alkalinity in the lake, in the open well, and in the bore well. Red basically means it's bad. Orange is slightly better. So this is alkaline water. Bagalur Lake is the first lake that gets this treated water. Then the water in the lake, after it sits there for like 2-3 days, gets pumped to another lake, and then from there it just percolates through gravity to multiple other lakes. Okay, so what we saw is that as you keep going through this lake chain, the water becomes better and better and better in its purity, because now nature itself is taking a part. It's not just dependent on humans and our STP systems trying to clear up this water. Next slide. So here are two opposing sides, right? Click on this. Click. On one side, it says, this stuff is too reactive. You don't have water. Now you go around putting dirty water everywhere. Okay, it's treated wastewater, of course. But do we really have that kind of trust in our government systems to clean up this water properly every single day? Honestly, none. I don't think any of us has that kind of, while we love our governments and all that stuff, we don't have that kind of trust that these government systems will work beautifully every single day, right? So they are saying this.
Is it audible? Is it audible? I couldn't hear the voice.
Sorry, I got myself mute.
All right, okay. All right. I'll play it again.
So when did I mute myself accidentally? Which part of, okay, anyway, you can read what I'm saying anyway. So the chronology of ecosystems, if you knew, for example, that birds come when it rains, even though it may not be raining a lot, you see that, okay, there is some kind of seasonality to frogs croaking and some flowers blooming and the birds coming. Suddenly now you have water all the time. Aren't you completely changing the ecosystem? And is this ethical? Rural lands, all these people are also paying taxes for water. Why should they not be getting clean rain-fed water, but they are getting treated wastewater? Right? Is this ethical or not? Now, the pro side says these are districts that are desperate for water. You have climate change. Everything is messed up anyway. These guys don't have any water. Over the past 30 years, the city has basically drained all these towns of their water resources. Now everybody is desperate. Now the districts that were initially against this entire system are now for the entire system, because they are like, yeah, we need water. It doesn't matter if it is dirty or if it's treated wastewater from Bangalore. What we are able to see is that if we treat it either through gravity or through our own processes, now we have clean water, and we have clean water throughout the year. And if we are worried about the crops that are being grown because of this water, we are moving away from edible crops to inedible crops. Let's move away from, say, rice and wheat and carrots. Let's go to mulberries. Let's go to something else. Let's go to jute, things like that. It's had a huge positive effect. Any politician who now wants to run from these districts, they say, we will give you water. We will give you treated wastewater from Bangalore, but we promise you that it will be amazingly high quality. And it is ethical because the water that is tested is safe. So is it truly safe? Let's go to the next slide. Next slide. Yeah. When we tested the water multiple times, it was actually safe. We did not, other than that alkalinity that I showed you about, other than that, heavy metals are within permissible limits. Other things like BOD, COD, TDS, coliforms, etc., were all within safe parameters. Next slide. And in terms of impact assessments, we see that these particular same areas are now able to produce higher levels of vegetables and fruits. Next slide. They are able to produce greater numbers of animals because now you have water for these animals. You also have higher production of milk in every single of these districts. Next slide. You also have higher incomes from these farmers. All these guys are now gaining more money, which also means that they are no longer migrating out of their towns. They actually find that it is economically fiscal to stay in their towns instead of running to Bangalore and then not having enough jobs over here. So, which also means that communities in these towns are staying stronger than they were 30 years ago, because now you actually have people staying back and they have that ownership and that link and that bond with that land. Next slide. Just click again. Yeah. So all this, I started the story was because I was telling you about AMR. I started the story with that child who was really sick, and we were wondering why in the world is this child getting all these resistant infections at this young age when this child has not been exposed to antibiotics before. And this is the truth, as I said, in children as well as in adults. So where is this antimicrobial resistance coming from? So this whole story started because we wanted to test antibiotics in the water. So click. We do see there are antibiotics in these waters. So while there may not be very high levels of other pollutants, because those have actually been removed by the treatment plants, because those treatment plants have been designed to remove these pollutants. However, no sewage treatment plant thus far actually has processes to remove antibiotics. While we see fairly low levels, like things like fluconazole, like linezolid, etc., these are antibiotics with low half-lives anyway. They automatically degrade when exposed to water or sunlight. Their half-lives are pretty low. But antibiotics with higher half-lives, which are very stable, for example, like isoniazid. Isoniazid is an anti-tubercular drug. It is actually given as a second-line TB medicine, and now actually nowadays, in the past year or so, the treatment guidelines have changed. It is also given as a prophylactic. If there is a household with a TB patient, all the other members in that household are given isoniazid as a preventative. So those drugs are actually seen in fairly high concentrations in the water. So what does this mean? Does this mean that you actually are inducing certain levels of antibiotic resistance because you constantly have a low level of antibiotics sitting around in these environmental sources? Is that what it means? This is the next question that we are trying to answer. Next slide. In addition to all of this, we've talked about a lot about treated wastewater. Let's not forget, untreated wastewater are also being used across the board in all parts of the country, including Tamil Nadu, definitely in Karnataka, where people are just like, hey, we don't have water and we have sewage pits. let's use the wastewater that we are producing or our own communities are producing, and we are going to use this for irrigation of our farms. So these are actually, local untreated wastewater in farms that are growing mulberry leaves. We've also seen the same thing in farms that grow coriander and palak and methi. So many of the greens, when you when you don't cook your greens, please be careful, because many of these are actually being grown in farms that use untreated wastewater. But we don't know. I mean, we do know the pathogen contents are of this water is very high. We don't know what the antibiotic content of this is. Okay, next slide, please. So wastewater is emerging as potentially the only solution to critical water crisis. Oh, thank you. Wastewater could be a method of finding AMR hotspots within a city, because you can do the same thing, right? In a city, like when you go to all your open drains and you start looking at the antibiotics in those open drains, you can also find out which of these areas in the city actually have high levels of antibiotics. And then that can tell you which are the hospitals nearby, because certain antibiotics can only be used in a hospital in an IV setting, not as oral medicines. Carbapenems, for example, you might have heard of if you've ever had the misfortune of having a relative in a hospital. You might have noticed that one of the first drugs that they give through the IV line is a drug called meropenem. Sorry, what's going on? Huh. So that meropenem can only be given by, as a carbapenem, can only be given by IV. If you're seeing meropenem resistant or pathogens in an open drain in your city, it basically means there is some hospital somewhere that is not treating its sewage properly. So it could be a method of finding out AMR hotspots and also finding out potential abusers and incorrect disposers of antibiotics. But wastewater could also potentially be a conduit of AMR, is what we are seeing. And the last couple of slides are what I'm going to talk about now. So we know that AMR is a proxy for broader health perspective on health. I'm not going to take too much time because we only have 20 minutes. Let's move on. Just click. And yeah. So basically it's saying that, you could, all of us as biotechnologists talk about AMR from the point of view from the point of view from the pathogen. You're like, which pathogen has it? Which genes have it? How is this gene getting transferred? What we are not able to see from our restricted points of view is that who's actually interacting with the carriers of these pathogens. If wastewater, if soil, if air, all these are carriers of AMR, or in this case, wastewater. Who is interacting? Which are the animals that are drinking that water? Which are the humans that are interacting with that water? How is it transferring from that water to the human, and then from that human to, let's say, babies who are actually quite vulnerable? And then what are all the geographical terrain contributors to spread of AMR? These are things that as biotechnologists, we don't think about. And I think it is time that we started thinking about it. We don't have to know all the answers, but we do have to work with other people who might be thinking about these things. So if you have a hydrologist or a geologist or an urban planner in your circles, start interacting with them. Because what does true faculty development mean? Faculty development does not mean that you become an amazing faculty in just your biotechnology field. It also means how are you now able to communicate with other scientists, right? Next slide. So this is what I was talking about, transdisciplinarity. As leaders, we are all scientists here. We are all been taught certain things that other people don't know. And we are privileged in that, right? How many of us actually, what percentage of the population actually ends up going to college? And of that, how many of us are able to do masters or PhDs? It's a very, very, very small proportion of the total population. And as such, we have a responsibility to talk to other people, not just other scientists, but we have a responsibility to talk to our communities, to businesses, to NGOs, to the government, to other people. And so this is where I want to stress that the 2 or 3 projects that I've told you about over here are transdisciplinary, but we need everyone to be a transdisciplinary thinker. Next slide. This is basically a glimpse of all the players that actually come out and have an influence. This can be scary, but I think as leaders and scientists, we should be working with them. In the projects that I've told you about, we had central government, state government, local government institutions, Indian for-profit companies, Indian NGOs, foreign profit, blah, 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 all these things. And every single of these, there are implications for money, and there are implications for data, and there are implications for ethics of all of these. Next slide. So I'm going to end up over here. I'm going to I think go on to the next slide. Enough. Boring. Next slide. This is one last slide I want to leave with. This is a slide showing, so these are all people who have come because of climate change and lack of water in their various states. And they've come to, in this case, it was Bombay or Pune. So this is basically in Maharashtra. Most of these have come to Bombay and Pune. These are also the same people who have come here looking for work and have died in these cities. So this was basically a mapping of deaths of migrant workers who have left their hometowns because of water scarcity. All right. So when you think about it, now you actually see like real world impacts, right? We are not just talking about some, airy-fairy theoretical situation. This is water scarcity is something that all of us are dealing with in our own ways, but it's also tragic in so many other people's lives, because it's also ending up in distress migration where they have to leave their places and come to larger cities. And because they are basically, there are thousands and thousands and thousands of these migrants, their lives are cheap and they end up in doing dirty, dangerous, difficult jobs, and many of them die. So I'm going to leave you with that. Just go to the next slide. I think I'm basically done.